you all, the organizers, as well as Sam and everybody for coordinating this meeting. This is virtual. The virtual meeting is a great podium to still connect with people in this COVID pandemic. Listening to all the talks in the morning, I've been more than inspired as a bench scientist to go back to the bench, and I'm hoping that can happen very soon. So today I will speak about drugs and targets in fibrosis from a preclinical perspective. Fibrosis occurs in several organs in the body: the skin, the bone, the heart, liver, lung, pancreas. Uh, so it's a universal phenomenon in biology where something goes awry with wound healing and you will have fibrosis happening. So looking at it from a preclinical perspective and at the drug discovery funnel, you start to realize, how do I start drug discovery? So drug discovery starts with an idea. It starts with identification of a target. And then you have to validate this target. You have to provide proof of biology, figure out a bioassay to set up screening, and then generate a lead molecule. So once you do that and you have lead optimization, then you can go to a first-in-human dose preparation. So the initial portion of preclinical drug discovery and development is extremely essential and is the most difficult part of drug discovery because most of the drugs fail at this juncture. So I'm just going to go into the steps in early discovery. The first and the foremost step is to get an idea for a drug target. And then you have to develop a bioassay for this target to prove that this is your proof of biology, this works. And once that happens, then you start to screen and develop a screening assay, develop a PD assay, develop a binding assay. And so all of this falls under this pharmacology regime. After that, you'd establish whether this drug is effective. So you go to PK, you do DMPK, and then you look at toxicity in a preclinical model, and you also look at efficacy in a preclinical model. So in a rodent model, before you can actually even approach an investigational new drug status. So I'm going to look and more concentrate on the idea of getting into the idea of a drug target in this presentation, with fibrosis, specifically cardiac fibrosis, as my topic of interest. So fibrosis is a well-recognized cause of morbidity and mortality. You can see from the slide that liver is the most organ that is most perfused, therefore it can develop most fibrosis, or very predominant number of fibrotic tissue is in the liver. But the heart is third in the paradigm. It is 1 in 60 of 1 in 60 patients will develop cardiac fibrosis. And that is a passion. Cardiovascular biology is my passion. And so if a patient has myocardial infarction or hypertension, aortic stenosis, hypertrophic cardiomyopathy, or heart failure, the propensity to develop cardiac fibrosis is very, very high. Looking at the history of cardiac fibrosis research, you can see initially that there were very low research papers available to show anything in fibrosis. Starting 1995 to 2015 and 2020, in the last 15 years, this has been recognized as a field that needs to be investigated. And still you can see that the papers that show are mostly biophysical signaling papers. You don't really have that much drug discovery, or you don't, even though there is drug discovery happening, there is no outcome happening. So what is cardiac fibrosis? Cardiac fibrosis is the pathological extracellular matrix remodeling, which leads to abnormalities in the matrix composition and quality. In the simplest format, ECM deposition is a protective mechanism that can be beneficial for wound healing and tissue regeneration. However, excessive ECM deposition, particularly collagen, leads to impaired tissue function, disease compliance, and leads to arrhythmias. Surplus collagen by activated fibroblasts, which are known as myofibroblasts, increases the stiffness of the wall and impedes normal systolic and diastolic mechanical function. And in today's world, HFpEF is the mantra, and HFpEF is fundamentally impacted by fibrosis. So when you look at, I've been reading this Nature paper, which impressed me a lot. What is the expressway to the core of fibrosis? So you have to find pathways when you're trying to do a drug discovery program that impact fibrosis. So there are two ways you can tackle this. One is to find a core pathway that is relevant in different organs and that can become fibrotic to be able to discover molecules that will serve as therapeutic targets. Another way is to look at regulatory pathways that influence this core pathway, but do not directly convert the initial stimulus in the basic component of fibrosis. So since organ speciation predates mammalian specialization, it provides a practical approach to uncover core and regulatory pathways if we go forward and start looking at these core pathways. We can also look at the different genes that have been identified and put together a Venn diagram of, coordinating cardiac fibrosis, liver fibrosis, and find those genes of importance that center in that Venn diagram and that tell us these are the predominant genes and go path forward from there. So in cardiac fibrosis, you can specifically see that the cardiomyocyte is 75% of the cardiomyocyte structural space, but 25% of the myocardial structural space is the cardiac interstitium that composes of the endothelial cells, the vascular smooth muscle cells, the macrophages, the mast cells, and the fibroblasts. And in reactive fibrosis, when hypertension, valvular disorders, diabetes, genetic composition or aging, you have reactive interstitial fibrosis that progresses towards necrosis and myocyte necrosis and myocardial infarction, which then causes infiltrative interstitial fibrosis. And you have infiltration, and you can see amyloidosis happening, and you have further cardiomyopathies. This then goes into replacement or scarring fibrosis, which is permanent and irreversible. And so then this is a portion where you have NYHA class 3 or 4 patients in cardiac myopathies where they have severe scarring and severe fibrosis, and those cardiac myocytes are irreparable. So as you can see, that fibrosis is a huge market for any company, and fibrosis is something that is very, very hard to tackle. So when you look at the mechanism of fibrosis, you see that there is a pathological stimuli that happens in the form of an insult. In the case of heart, it can be hypertension, atherosclerosis, and then consistently which lead to clotting or further on myocardial infarction, which then activate the fibroblasts to become myofibroblasts. You have an increase in the calcium within the cell, which then releases MMPs. Then you have ECM happening. Then you have the cytokines that come out, which is TGFbeta, IL-6, and a lot of the other inflammatory cytokines. Then these act as regulatory pathological stimuli and forms a cycle wherein you have further fibrosis happening, which then leads to diseases such as arrhythmia, hypertrophy, and heart failure. So what can we do? So then you start looking at the regulatory factors, one of them being MMPs. And so you look at the MMP12 maybe and start saying, oh, can I use this as a target to inhibit this pathway and prevent fibrosis from happening? MMP wears multiple hats. It is used for wound healing, removal of necrotic cells. It reduces neutrophils, reduces chemotaxis, and improves or increases the anti-inflammatory cytokines. So it has a beneficial effect. However, it also activates the fibroblasts, increase TGF-beta activity, and then causes fibrosis. So if something goes awry in this wound healing process, then you have fibroblast activation and you have increase in fibrosis. So the trials that have happened with MMP inhibitors have therefore not worked. So then you start to look at the fibroblast biomarkers. Although small but nascent, there's a consistent group of fibroblast biomarkers that are now being discovered and adapted every single day. And then you can see that these are periostin, vimentin, fibronectin, ectodisplacin or the PDGF receptor, alpha smooth muscle actin, THY1, which is also known as CD90, which is a membrane glycoprotein. People are also working on TMP activation and also suppressing the immune system as a medium of trying to reduce fibrosis. So these are all the different targets that are being looked at because fibrosis is a disease which is multifactorial. So what are the current therapeutic approaches that are present? Since there is a strong clinical and economic sense for developing new effective therapeutic approaches, the anti-fibrotic therapy starts at TGF-beta. So there are TGF-beta pathway inhibitors, MMP inhibitors, inhibitors of mast cell, MCP1, CXCL2 cytokines. The newer field is the miRNA, which I will talk about a little bit. Some of the biomaterial therapies, cell therapy with FAP proteins, and then reprogramming of SEVP and proteins such as serelaxin and periostin, and the communication, the gap junction proteins such as connexin 43 and GAP26. So from the current therapeutic approaches, TGF-beta, which is a very, very well-known cytokine that actually moves towards causing fibrosis, you can see that it's present in all kinds of fibrosis, in renal fibrosis, liver fibrosis, or the cardiac fibrosis. It increases the myofibroblast, ECM production, fibroblast proliferation, cell immigration, and scar formation. But TGF-beta is also required for wound healing. So this is a very perplexing question to any scientist. What is that point at which you stop the signal? And that is a multi-million-dollar question. If this can be answered, then we have a solution for fibrosis. So I put it out there to all researchers in the field to think of that, because you have this going haywire and then it can go in two different directions. One, it can be tissue fibrosis, and in the other organs where tumorigenesis is possible, it can lead towards tumor or carcinomas. Another therapeutic is Pamrevir, which is a fully human monoclonal antibody, which is against connective tissue growth factors. This has been used in DMD patients, that is the Duchenne muscular dystrophy patients. It has shown improvement in cardiac fibrosis by improving the left ventricular ejection fraction and reduction in cardiac fibrosis, as you can see from this graph here. Another current therapeutic that is very popular and being done, a lot of research is being carried out of this, is the miRNAs, which are critical regulators of cardiovascular function. And specifically miRNA 21, which is found to be directly targeted to KBTBD7, which promotes the DAM-triggered inflammatory responses in macrophages. MIR21 attenuates inflammation, cardiac dysfunction, suggesting that it might be a novel potential therapeutic for MI. So when you look at this, you can see that miRNA21 deficient mice had worse functional deficiencies as compared the knockout mice had worse deficiencies as compared to the wild type mice. So improving or increasing the miRNA21 is a great solution for this particular disease. However, how this can be modulated or how anti-fibrotic activity with miRNAs as a therapeutic is still under investigational situations, because remodeling and at what point should the therapy be given is a question that is, therefore it can have limited potential. So this understanding fibrosis in detail and going to the molecular mechanisms of fibrosis becomes very important at an organ level. Another current therapeutic approach that I am really keen about and interested is the biomaterial assisted therapies. In biomaterial assisted therapies, you have scaffolds that provide biochemical stimuli. They act as ECM or replacement ECM and are significant and are of great significance, especially in hydrogel therapies. So while using hydrogels to decrease cardiac fibrosis, the time of injection affects the therapeutic efficiency. And the wall stress gradually increases from the necrotic phase to the fibrotic phase. So it's extremely important to find a time when the hydrogel can be delivered into the heart. And this can be done by using a endocardial injection catheter in the LV, and you can use the Ventrigels such as porcine cardiac ECM hydrogels. They all have their own complications. However, the timing of the hydrogel injection should be considered in order to achieve optimal therapeutic effects. And in trials, in early trials, which are about 8-week trials, you have seen reduced cardiac fibrosis and enhanced vascularization, which significantly increase cardiac function. In a six, in a, in the, I mean, decrease in the NYHA class, meaning that patients who were in class 2 fibrosis have come back to class 1 fibrosis. Even in there has been LV remodeling in the late group, which is the late NYHA class 3 patients. So even 1 year post MI versus less than 1 year post MI, you have seen ventricular remodeling and better ejection fractions. Immunotherapy is a fad today for most diseases. And as usual, using the FAP protein, FAP CAR T cells, it's possible to target cardiac fibrosis, and this has been seen in mice with engineered T cells and immunotherapy. The fibroblast activation protein, FAP1, is an integral membrane gelatinase that belongs to the serine protease family. When FAP1 is used to distinguish fibroblasts from other cell types, it is present in melanocytes. And FAP1 can be considered to be a fibroblast specific in the myocardium, but it does not but not across the whole body. So this specificity is what confers its attractiveness as a target. So adoptive transfer of the T cells that express the chimeric antigen receptor against the fibroblast activation protein results in a significant reduction in cardiac fibrosis and restoration of function of injury in mice. The results provide a proof of principle for the development of immunotherapeutic drugs for the treatment of cardiac disease. However, there are limitations for this therapy. Uh, Continued investigation to identify unique antigens that are expressed by activated cardiac fibroblasts may identify alternative age antigens, combination of antigens that might be effective targets in this therapy. With any T cell therapy, off-target effects can range from mild to serious. So it might be necessary to engineer modified T cells with a kill switch to limit the time of survival to minimize any off-target side effects. Another therapeutic possibility is the fibroblast reprogramming. Sendai virus vectors that express cardiac reprogramming factors efficiently reprogram both mouse and human fibroblasts into integration-free ICMs in vitro. This is a Nobel Prize-winning technology that was discovered by Yamanka and their colleagues, who reported the generation of iPSCs from somatic cells by retrovirus-mediated overexpression of OTC4, SOX2, Krupel-like factor 4, and MYC. So if you can reprogram cells into defined phenotypes without reverting to the intervening stem cell. So this is an exciting technology, and if we can leverage this technology, then microRNAs and actin-binding peptide, thymosin B4, etc., can be reprogrammed into cardiac fibroblasts and into cardiac myocytes. And using these Sendai virus vectors, this can be achieved. So it has also been shown that using the Sendai virus vectors, that there is a decrease in fibrosis, as you can see in the decrease in collagen 1 here in this staining. And also in this echo, where you can see that the Sendai virus GMT, you have better echo performance with the transfected in mice. So last but not the least, I'm going to end this presentation with a question. You have acute injury, you have chronic inflammation, you have autoimmune disorder, you have genetic disorders. All of these alter cytokines, cause ECM changes, and lead to fibrosis in all of these multiplicity of organs in the human body. If you remove the stimulus at this point, then you start to see regression in fibrosis. So what is the next for fibrosis therapy? What can you really achieve? Is it prevention of chronic inflammation? Is it mitigation of by inhibition of the stimuli, or can we go back to regenerative medicines? Or can all of these be approached at different stages of fibrosis? Has this to be done differently per organ, or can we find a unified mechanism for fibrosis? This is the core question that we should answer in order to develop new targets for fibrosis. Thank you. And I'm open for questions.
Thank you very much. That was a very insightful presentation. As the questions come in, I have one to start, and that is the previous products that were developed for fibrosis that failed, didn't work. You think it's because they were administered too late, meaning that the diagnosis was made too late and the fibrosis had progressed too far? Or is it possible that the treatment period for the fibrosis was not long enough and it might take a year or more in order to see changes? Do you have any thoughts on that?
So my personal understanding is that I think that it is the initiation. It is when you start treating the fibrosis. It's identification of that wound healing going pathological. So I think if you're able to remove that stimulus, or if you're able to mitigate that stimulus, that would be complete reversal of the fibrosis. If you're going to treat fibrotic tissue, which is established fibrosis over a period of time, and if you still treat it for maybe 2 years or 3 years, I don't think the length of the treatment will provide enough regeneration.
Okay, thank you. We have a question from Hugo. The question is, has there been single cell studies that would highlight specific cell types that would regulate tissue going to fibrosis versus normal versus tumor?
There have been some studies specifically with TGFbeta looking at single cells, and I can find those papers and send them to you. I think there have been some studies that have been done, but they've all been at an academic level.
Okay. Another question, this one is from Laura Prendergast. The dichotomous outcomes, fibrosis versus tumorigenesis, are interesting. If I were looking for a regulatory factor, I would look into stem cells. Is anyone investigating that?
There are a few companies that are investigating stem cells. I am not a specialist in stem cells. However, from literature and from market intelligence, definitely it's a very, very exciting field to be in. And I think they can provide solutions to a lot of diseases. So stem cells, yes.
Okay. Another question is from Dr. Josh Atiba. How does fibrosis affect CNS and heart and kidney as related to amyloid deposits?
So as related to amyloid deposits, I think the central nervous system and the heart are very similar in terms of the amyloidosis pattern of cells that develop. However, in every organ, the cell types are different. The proteins that are expressed in the cell types may be different. So you have the ability to target those specific proteins or the genes that confer that. However, it's important to understand when I look at it in terms of fibrosis or when that's happening, it's the initial stimulus that pushes that fibrosis to happen and divide into XYZ cell types and confer amyloidosis. So that's something that has always been perplexing, and that's something that is part of my research very intensively, trying to understand what is that stimulus that pushes fibrosis, and then it goes, I mean, if you go to the liver, for example, or the kidney or the heart, or like you mentioned, the CNS, then the fibrosis patterns start to differ. You start to see here that you have hepatocytes or the endothelium. You start to see ECM formation. The end result is still the same, necrosis, and then you have a lot of matrix proteins, and you have cell death. So it's the in between. It's like there is a stimulus, and then the cells do their job, and then you have an endpoint. So that is where, I mean, the common factor is the initial portion. So we have to look at amyloidosis from where the stimulus begins.
Okay, thank you very much, Raja, and we wish you continued success with your research.
Thank you.
Thank you. Thank you.